All right, we'll get started soon. Hey. You're taking AP Chem at school?
Yeah, because there was like too much with my
Going back for it. I see. Great to have you. Well, I'm just graphing this while waiting. This is you get to learn about memories of bees. So fun. All the crazy stuff in the nature. Hey guys. Okay, so as before, we still have all the class notes in video format. So in case you need to review it. So what Office did, I just want to show you in case you haven't check it out into this. Under what is it? Under in class. No, under what is this? Right here, class recording. Right, you can see under class recording. This was last week, test number 3, February 22nd. So you can look at under here the notes, and also the recording. So if you click on the recording, you can download the recording, because some of the stuff, the answers, I did not write it out because it's kind of really repetitive when we said it so many times to write it out, but I'll try to write out as much as I can. So anyways, I just want to let you know that. Okay, so if you need to review some of the stuff that we said, please welcome to go back to the meeting notes. Okay, all right, so for those you are home, let's pay attention so we can get this done. Yeah, okay. Give me a sec. Okay, we'll get started. People having problem coming in. So let's see. So also, if you can't find the Zoom link, they put it out here for classroom link. That way, it won't disappear. Okay. Okay, maybe not. Where's the Zoom link? What the heck? I thought I was just at the Zoom link. Let's see. One sec. Okay, sorry. So, you guys ready? Right? Right. You guys worried about the war, Russia and Ukraine? Yeah, some of you guys are. It's unthinkable something like this could happen in this age, right? Yeah, and then doesn't seem like this is what the problem is with the autocrat like Putin. Okay, his will against everybody else's, and nothing a lot of Russian can about it. We have seen. Never imagine I will, even in my lifetime, will see another modern warfare like this. And Russia has more nuclear weapons than United States. They rank number 1 in nuclear capacity weaponry. So, so basically, he's holding the entire world hostage, betting on the world won't do anything militarily to intervene. So, all right. So, treasure what you have. We're very lucky to be in the United States. All right. So I was so this is the first question, guys. Okay. Let's see. So the first question here is about memories of bees. I just kind of started to plot while we were waiting. But again, to review, your first two questions will be about 10 points. And this one here, so sometimes on these questions, you got to always remember there's always clues in the questions about specific knowledges that you need to know, background informations that sometimes that you need. Sometimes they feel like they're redundant, but sometimes they're good sources of answering the question. Yeah. Okay, so this is the question. So if you look at this one here, we were told that in flowering plants here, okay, so flowering plants are the most accessible of all plants because ability to have flowers to attract pollinations, they can gain genetic variations that way. And being able to have flowers and bear fruit also helps with the dispersal of their seeds to distant places so that they don't self-compete. So most diverse. Insects would be the most diverse for animals. Yeah. Okay, so it tells you what pollination is, so important, right? It's a fertilization of an egg with a sperm, right? So when egg is fertilized, it becomes a seed, and the ovary of the flower becomes the fruit. So the entire flower is a reproductive structure. Next time you sniff a flower, there's male and female reproductive parts, right? So the reason they're scent is because they attract insects, they attract mammals like bats, they also attract birds, right? So to help them pollination and also seed dispersal. All right. So it says here they use chemicals for sure, right? And when a bee visits a flower, in addition to transfer pollen, bees get nectar. So this is kind of like a mutualism, right? Bees get food, nectar, and then they can make honey, right? Nectar contains sugar, but certain plants also produce caffeines. So like I love caffeines. I drink caffeines every day. I don't get addicted to it, but if you guys never had caffeine, it's probably you need it to get through college. Yeah. Okay. So caffeine is bitter tasting compound. For some reason, people like it. Okay. Can be toxic at high concentration. That's important. So too high is not good. Too high of anything is not good. Yeah. You cannot just pop in vitamins because it's good for your body. Overdose vitamins can also have bad health effects. So to investigate the role of caffeine, so here's the point of the question. After all that background story, they want you to know the role of caffeine in nectar. Why do bees put caffeines in the nectar, right? And the researchers studied the effect of 0.1 millimolar caffeine on the behavior of the bees. And what behavior? So finally, the last sentence talk about the bee's memory of a nectar source. So if you feed the bees a drug like caffeine, does it get the bee addicted to you so that it comes back to your flower, this flower, more regularly, right? So this is the bee's memory. So if you look at this, so this is 10 minutes. So initially when you look at this, what the heck, 10 minutes memory? 24 hours, minutes. Then the keyword here is revisiting, right? So revisiting after 10 minutes, revisiting the same nectar, the same flower after 24 hours. So it's about coming back to the same flower again if you were to use caffeine. Control is probably no caffeine, right? So you can see that, this is the short-term memory, 10 minutes. Maybe the bee remembered pretty well. Okay, maybe 24 hours, look at the control, it dropped, right? From 72 down to 0.41 probability. Okay, 41% revisiting the same nectar source after 24 hours. So that's what the data is trying to say. And it seems like caffeine helps with long-term memory because there is a increase, right? Almost double. And I don't think it helps with short-term memory. But every time you're given the standard errors of the mean, you have to make sure that the standard error of the means do not overlap when they do overlap, the results are similar and they are not different significantly, statistically speaking, right? But if the bars do not overlap, then the result shows a significant difference statistically. So that's all that means. Okay, so I was graphing the first question because the first question, for those kind of graphing questions, are really easy, but graphing it together kind of help us to figure out what to do. So on the axis provide, construct a labeled graph. Usually labeled graph is like 3 points. Yeah, you have to do the axis with units. You have to do the bars. And then in this case, the error bars, the standard error of the mean also has points. So, and then I want you to describe the effect of the caffeine. Okay, so let me take a picture of this so that we can move it over. And then part C wants you to design an experiment. Now remember, design an experiment, you have to talk about control and controlled variables. What else must be held constant in order for you to see your experimental variable? So you have to talk about maybe your hypothesis or no hypothesis. You've got to predict the results if your no hypothesis is true or if it's false, right? So you're not actually going to do it, but you have to write enough for the grader to convince that if you were actually to do the experiment on your own, you know what to do and know what the results mean. So it is definitely an important type of question that you'll see on AP Bio. And especially with the first two, design an experiment, interpreting experiment is a very important skill. So I was doing this while we were waiting. So what I was looking at here is the y-axis is the probability, and I was looking at if I have the highest one is 0.8, so I begin to interval out my y-axis from 0.1, 0.2 to 0.9, and that seems to work. And I label the y-axis unit exactly the same way as given. So don't come up with your own term. Just use whatever they give you. Yeah, I wouldn't even abbreviate like what I'm doing here on the test. Just write out whatever you see. And the x-axis, I was just doing that. So this would be the memory. Like that, right? 10 minutes, 24 hours. And after I draw in the so when I draw this, when I draw the arrow bars. When I draw the bar graph, I usually draw this first. For example, if I read 0.72, and I'll go 0.72 like this. I know it's a little bit below 0.75, so I draw a line like that. And then 0.83, so I go to 0.83 like right here. I just draw the horizontal line. And then I go to 0.41, so a little bit after. And then I go to 0.78, so a little bit below, right? And then I draw the vertical lines to connect them. Does that make sense? Yeah? It's amazing how I can draw these vertical lines on the digital pad. It takes a lot of skill, trust me. If you try digital pads, it's not easy to draw a straight line. Okay, it's very difficult to write your own name on these things. Yeah, but so I draw like this, and then I go after the arrow bars. So how do I draw the arrow bars? Well, the first one, for example, says 0.09. So I actually add 0.09, like 0.72 plus minus 0.09. So I know it's 0.81. And then if I subtract, will be 7, 63, right? 0.63, right? So I know it has to be between here. So I go to 0.63 down here, I go to 0.81 up here, and then I just draw a line in between. That's what I do, yeah? So you literally add and subtract, so you know what your range for the arrow bar is. And the next one, for example, is 0.83. Plus minus 0.07. So if you plus 0.07, that's 0.9. And if you minus 0.07, that will be 0.76, right? So I know it's from 0.76 right here, a little bit above. And then 0.9 is up here. And then I just connect them. So that's that, okay? So anyway, I was working on this. And the last one was 0.78. So that would be 86 and 0.7. So 0.7 and 0.86, a little bit higher. So there. So from here, I see that our initial assumption that there is no difference between the control for the 10 minutes and the caffeine for the 10 minutes. That caffeine did not produce a difference in the short-term memory of bees. because although it seems like there is 0.72 to 0.83, but the error bars overlap, right? Whenever the error bars overlap, you have to believe that your two results are not significantly different. They're similar, in other words, okay? But long-term memory, our initial assumption was right. Error bars do not overlap, that caffeine does improve long-term memory of bees revisiting a particular flower. And is evident here after 24 hours is considered long-term, okay? Like that. And then this is true for humans as well. I don't know how much caffeine helps, but if you try to study and memorize a whole bunch of vocabularies the night before a test, in the morning you'll forget a lot of them. But if you just freshen up a little bit, like maybe 30 minutes before the test, your memory will jump. Yeah, so definitely do that for any particular test that rely on memory. Okay. Alrighty. So describe the effect of caffeine on the short term, long term. We were just talking about it, right? So, and we got to be able to put that in words. Again, when you kind of understand this, when people talking about it, it's always easier than if you were to do it on your own. So definitely, I would encourage you, right, to try to type it up on your own. We don't have time for you guys to read out some of your answers. I used to do that. Maybe we can do that when everybody is in person. I was hoping maybe the last day of AP we can do it in person, so I don't have to use these digital stuff. Okay, I kind of like it, but I also don't. I don't know. So, but here, short-term memory effect of describe. So description means it doesn't have to be very long. Yeah. So caffeine. So I'm gonna say caffeine has no effect, okay, on short-term memory of these. Yeah. Okay. And then now, do I have to justify? The justification is the error bars. It doesn't ask you to justify, but I don't know. I usually want to, because I know the answer, I want to just make sure I get the point as the date, as the 10 minutes error bars overlap, suggesting similar results. Yeah. Okay. So it doesn't take you very long to just add that. Okay. And then secondly, caffeine improves. Now, some people said description. Should I use data? Should I use number? Because that crossed my mind. I want to use it because before there's no difference, so I'm not going to use it. But right now, for the 24 hours, there is improvement, right? It went from 0.41 to 0.78. Yeah. So I want to do that. That to me is a better description than not using any numbers when you're given. So if you're able to use numbers because describe, the more detail, the better, right? So caffeine improves these long-term memory as data shown from 0.41 to 0.78, was it? Yeah. So 0.41 to 0.78, right? Okay. Caffeine does improve these long-term memory, right? Okay. So all right, like that. Okay. Next one. So easy points here. Just make sure you don't overdo and waste time, and make sure you move on quickly. So you save time for more difficult question. Yeah. Okay. So now the second part, the part C is probably the most difficult one because if you have to design an experiment, you got to think about it a little bit. So you have to look for what exactly I'm trying to prove. So you know hypothesis or hypothesis, right? When you design an experiment. Okay. So the question is that design an experiment using artificial flower. Again, that is to minimize. What that means is to control your other variables. Okay, artificial flower. Okay, so every one of them is identical. All right, and then what? What are we supposed to do here? So take one of. All right. Okay. Potential of increasing caffeine. concentrations, right? So you put higher and higher doses of caffeine on the number of floral visits, right? So we know caffeine addicts the bees to visit them, but what if you use too high, right? Maybe the bees don't like it anymore, right? So that's why you're trying to investigate. So design the experiment, okay? So identify the null hypothesis. Again, null hypothesis would be that increasing caffeine concentration has no effect. Remember, null hypothesis is that you're not going to see anything different from your experimental variable. In this case, it's increasing caffeine concentration. There's not going to be any difference in the negative effect. Yeah? And also, they specifically ask for control and your predicted results. That can be, so your result will be used to reject, yeah, no hypothesis. So you ask at least to do 1, 2, 3. 3 things at least. So design this experiment, right? Okay, so again, we talked about the null hypothesis, but again, to type it out is not easy, right? But if you look at the scoring guide, I also pulled out the scoring guide so we can go back and forth, and you can see that the scoring guide here was this one. So before was 3 points, right, for graph. So give yourself 3 points for graphing them, please, for part A. And part B is just the 2 statement. You can see that caffeine does not affect short-term memory at 10 minutes. It doesn't even talk about the error bars. That would be good enough for AP grader. And the second one, caffeine does increase the long-term memory. That's all. It does not put in numbers. So they will take the bare minimum, I suppose, yeah? Okay, so here's the designing the experiment. So let's look at what they put down for null hypothesis, right? So null hypothesis here says increasing caffeine concentration has no effect. And I will elaborate, no effect on what? The number of floral visit by bees, because these are the exact wording, you see? Negative effect on increasing caffeine on what? On the number of floral visit by bees. So all they did is repeat that. Okay. Now remember, this is no hypothesis. Not your hypothesis. If it's my hypothesis, if I were to put a hypothesis, I would say that yeah, increases the memory, yeah, increases the number of visits until a certain threshold when caffeine becomes toxic. That would be my hypothesis. But that is not the null hypothesis. Okay. All right. So that's one point. Very easy. Write that statement out. Right. Second part. What will be the control, right? So you think about the control will be no caffeine, right? Because you have the caffeine or no caffeine or low caffeine. So because you want to see the increasing effect. So let's see what they put down for the control, okay? So the control here will be no caffeine. Okay, your artificial flowers, there's nectar, but there's no caffeine inside. And then the other ones are your experiment. So you put 0.1 millimolar, 0.2, 0.3, 0.4, and then you want to see how many floral visits, right? Okay, so what must be controlled variables? What variables must be held constant? Well, we have the bees, right? You cannot have a group of bees that is old, that doesn't really fly anymore. And then the other group using young and healthy bees. So the number of bees, the age of the bees, the duration, right? You want to test this, the temperature, right? The amount of water present, all the other experimental conditions must be held constant for every single one of your group. You cannot favor the 0.1 molar and have them have more nectar in the flowers. And the 0.9 molar has very few nectars, right? So that wouldn't work. So everything other than the caffeine concentration must be held constant in every one of your experimental groups. So that is a very important condition, right? You cannot have the bees distracted by noise, distracted by predator, by the amount of sunlight or the temperature differences in your experiment. So that's what control tells you. So everything must be identical. I would actually include that, but all identical condition, but the one that has no caffeine only. All right, so that is that one. So 1 point. And then finally, they ask you to predict, right? Your results. Results to do what? To reject. You can see that we are trying to reject the null hypothesis. So null hypothesis says that there's no effect on caffeine. Well, I mean, if caffeine has no effect on the probability of visiting the flower. So your predicted result is that there is an effect that increasing dosage of caffeine improves the number of visits or increases the number of floor visits or decrease. As long as they're different and allows you to reject your null hypothesis, that would be great. Yeah. So your result, your predicted result has to be different from your control. Yeah. So I will put down that increasing. Let me just put that one in. Yeah. Where is the note? Because this one, let's see. Put that in here. Okay. So increasing caffeine. Caffeine concentration does well increases. How about that? And I'm trying to use the same wording. Number of visits by bees. So that will allows me to reject my original null hypothesis that it doesn't. Okay. So any questions on this? Yeah. Again, they could extend the question and ask you stuff about what conditions must be how constant, how do you improve your control, right? So if for example, if the student says this is my control, I don't like this control, so how do I improve it, right? So this is the control by student. Okay, so it would be a wildflower, okay, with nectar and no caffeine. So how would this control be improved? How do I improve it, right? So I try to word it in such a way so that this is actual flower. That's not good. That will control additional, this will bring additional variables. So a better one would be an artificial flower, just like the rest of the, an artificial flower with nectar, okay? And no caffeine, yeah? So this would be the same as the experimental groups, same flowers as the experimental groups, right? So you can come up with, they can come up with questions by tweaking it a little bit and for you to justify your control, yeah? Okay, all right. So that it just depends what they want, okay? What the focus is. So just keep that in mind. So every question like this experiment, you want to really think through so that if they do tweak them, you'll be okay. And the last one. Let's see what the last question is. We have researchers found that nectar with caffeine tend to have lower sugar content than nectars without. Okay, so if you have caffeine, you have less sugar, but it improves the bee's memory. I guess that's a trade-off, right? Okay, all right. So plants use less energy to produce caffeine than they do to produce sugar. Well, that's important. We have to be given that it's easier for a plant to make caffeine than to make a bunch of nectar sugar, right? So the question is, propose one benefit to plants. What is the benefit to plant that produce nectars with caffeine with a lower sugar content? So that's quite obvious, right? If you kind of just think about it, what is the benefit to plant to produce more caffeine and less sugar? Wouldn't that save plant energy, right? Because nectar takes energy, right? So the first thing came to my mind is energy, right? Just plant do that uses less energy and still get better revisitations. So that's the first thing popped to my mind. So let me, sometimes if you have a good idea pop to mind, you want to write it down as soon as you can before you start forgetting them. If I keep talking, I'll forget what I said. Yeah. So, okay, so here it is. And when you do this, I used to have a student who repeats everything. You don't want to waste too much time repeating it. Okay, so propose one benefit. So you don't want to say that one proposed benefit to a plant that produce nectar with caffeine. Oh my goodness, you just repeat the whole thing and waste like 30 seconds, right? And especially if you write slow. So I would just say that, you know, one benefit, okay, to plant, and that's it. Yeah, one benefit plant would be what was I saying? Would be energy conservation, or okay, while having more revisitation with caffeine, right? So they get a benefit of both, right? So that's it, yeah. And what is one cost to bee? Yeah. So the cost to bees, yeah. So they get addicted. They would visit the flower that give them less for doing more work, right? So you don't want to be that bee, yeah. But okay, so one cost. So there's a one cost to bees would be oops, to bees would be less. Well, you can say that one cost for the bees would be less sugar per visit. One cost would be less sugar per visit. Okay, making trips less efficient. Yeah. So you don't have to put into big words. I would just say that bees that visit flowers with lower sugar means that the bees have to make more trips or work harder to get the same amount of sugar for honey, right? So something like that would be okay. Just that you don't have to be restrained to one way of starting the particular answer. Okay, so let's see what the scoring guide says here. So plants benefit, more pollen is transferred because more visit by pollinators. Sure, that's why they use caffeine, right? And plants store energy and have more energy available for other uses. So they don't have to spend so much energy trying to get pollinated. Okay, and propose the cost of bees. So the individual bees would have to visit more flowers by using more energy, right? Okay, you can also talk about the bees as a colony, same thing. They will make less honey or lower quality honey, right? So there's a lot of ways you can answer this as long as you hit the right point. All right, let's move on to the next question. So that was so that is 2 points there, yeah. So please give yourself some points before we move on. So again, the graph is 3 points. Okay, you gotta have the bar graph correctly. You gotta have the error bars, and you have to label X and Y axis. And then describe is 1 point for short term, 1 point for long term. Design experiment is 3 points. We said there's 3 things to ask you. 1 for no hypothesis, increasing caffeine concentration and no effect on the floral visits, and 1 for control and 1 for any predicted result. I said that increasing caffeine does increase the number of floral visits, which is not what the control is showing, right? Then that would allows me to reject no hypothesis. And the last one benefit, 1 cost, 1 point each. All right, so please add them up. Let's look at the second question. Second question, see, we're given a graph. So here's the graph, and it's about, all right. So it says here, so we don't know what the graph means, yeah, but so you have to read this. So there's no point of reading this, okay, other than that seed germination is the y-axis, so percent of seed germination. So when seed germinates, they will burst out of the seed coat using cell respiration, and they will have to rupture out of the soil and then to start grow taller and taller, and then they will start to have foliages, leaves, and then the leaves will start turn green. That's called greening, and then they can do photosynthesis. But initially, the energy from seed germination comes from the seed reserve, by cellular respiration. Germinating seeds does not use photosynthesis. It's unable to do photosynthesis when there's no leaves. All right. Okay, and then under the ground too. So it's about seed germination. And I guess these compounds affect seed germination. So which compound have the greatest seed germination would be CAR, K-A-R, right? See, the percentage is the biggest, yeah? And then TMB on the bottom seems to do the opposite from control. I'm looking at the control, so I know what to compare to, right? CAR is higher than the control, and TMB is low. control. Axis is time. So we'll come back to that. So fire occur in some ecosystem. Right, fire is one of the disturbance. Yeah, one of the most important thing about ecosystem is called ecological succession. Yeah, and that's what that is when community changes over time. So ecological succession. Yeah, so fire would be a disturbance, a natural disaster that would actually alter the course of succession, right? The replacement of plant, animal, community over time. That's what ecological succession means. Succession is due to competition. Okay, usually you start with pioneer species occupy a barren land, and pioneer species are usually lichens, which is a mutualism between cyanobacteria and fungus. Yeah. Okay. So lichens will be replaced by small plants, the land dwelling, and then by larger bushes, and eventually bigger and bigger trees. Okay. So we're told about this thing here that fire destroyed everything above ground. Many species of plants, they respond to compounds in smoke that regulate seed germination after major fire. So we're talking about compounds in smoke that will cause the seed to germinate. Weird. So we have CAR and we have TMB. Don't try to pronounce them. Yeah. So the water-soluble compounds found in the smoke. So these are actual compounds in the smoke that are deposited in the soil after the fire. So CAR and TMB, what it did is that bind to the receptor protein in the seed. So the seed would bind to either CAR or TMB or both after a fire because they are deposited in the soil. And we said that we were looking at CAR, remember? We said that, okay, so what is CAR? And we knew that CAR helps germination. So the seeds that bind to KAR will germinate, and the seed that bind to TMB will germinate less, right? If you have 100 seeds, right? So that seems to be what they're doing. These compounds, one promotes and one inhibits seed germination. There must be a reason. In a study on the effect of smoke on seeds, researchers recorded the timing and the percent. So good. So we're told why we need the time. The timing is the x-axis, and the percent of germination is the y-axis. So I'm going to go back to look at the timing. As you can see, the timing is also different. So we also look at the CAR. You see the CAR start germinating 12 hours, and then control is around here, right? So control is later. And TMB alone germinate even later, right? So closer to 24. So not only CAR has greater percent of germination, okay, it also allows the plants to germinate earlier than the control, where TMB inhibits the total percent of germination and also delays germination. So there's two effects, the percent and the timing of germination. So how do we know that? We knew this because we're told. So there's always clue, yeah, for you to look for how to interpret the result. Okay, so now the various combinations of CAR, TMB are shown in figure 1, which is what we saw, okay? In the second investigation, into the effect of available water on seed germination after fire. So effect of water on seed germination. So do you need water to germinate? Common sense will tell you, yes, water for sure is required. It will soak, the seed has to be soaked in water, it's called imbibition, so that it softens the seed coat, allows the embryo to power out of it, right? So yes, you need water, amongst other things. So researchers treated the seeds with CAR and TMB, and the treated seeds were then divided into two experimental groups. One have water rinse, and the other has no water rinse. So you rinse with water, and the other group of seeds, there's no water. The seed were then incubated along with a group of controls that were not treated. So whatever that is, let's look at the data, okay? So the data is here. So this big, gigantic chart right here, yeah? So I'm just gonna keep it out maybe like this. So this is the results, okay? Data table, okay? So we have to somehow interpret the data table, I guess, Oh, yikes. I'm gonna make it a little bit smaller, hopefully. Okay. All right, so this is the data table, okay? And then this is the question that we have. All right, so let's see what we have to answer. So I'm not gonna read the data table and then just go right to the question, yeah? So here's some of the questions and see if we can type it out, okay? So the researcher for first question made the following claims about the effect of CAR and TMB on seed germination relative to the control. So this has to do with figure 1. It says on the bottom, provide support using the data from figure 1, okay? So my figure 1, okay, so let me go to the side. I don't have figure 1, but this is the question. So these ones are, I would imagine every bullet is 1 point. So make sure you're able to answer every bullet. And it is basically what we are talking about when we look at the graph here. This is figure 1, right? So the question is, the researcher made the following, right? So CAR alone affects the timing. And CAR alone affects the percent. So timing is the x-axis, right? And the percent would be the y-axis, okay? So this is part A. So this is 2A. So what do you guys think here, right? So 2A, you got all these bullets, right? So we're asked to do those, so we're on 2A, yeah? Okay, so what do you think? So CAR alone affects the timing, so they want you to use the data in figure 1, which is shown here, for each of the researchers' claim, right? So what would you say for this one? Because we are looking at CAR, and the timing is earlier, right? See, the control is later. So I'm actually going to see 1, 2, 3, 4. So it seems like there's 4 intervals, right? There's 1 interval, 2 intervals, 3 intervals, 4 intervals between 12 and 24. I want to know what each interval is, right? So you can subtract 24 minus 12 is 12, divided by 4 is 3. So this would be 15. So I have some numbers, right? So control started to germinate maybe 15, or this is 18, this is 21, right? So I have some numbers to look at. So each interval is 3 hours, in other words, okay? So because I'm looking at the timings compared to the control, right? So I need to use the data to support. So I want to justify support, okay? So CAR, so it's about the timing. Plant, seeds, okay, let me see, okay, so we go. Seeds with CAR started germination earlier around, and then control, yeah? And then, so 12 hours, okay, with CAR. And then I'm gonna say 15 hours with control. So I don't feel well unless I give some number from the data. So this one is directly from the data. So I'm trying to quote the data as much as possible. So if the question wants you to, use the data, then I am using the data, which is timing. Then I have to go after the x-axis. So sometimes we're not sure how much of data do we need to use. This one, I believe you do need the data. Yeah, so that makes a big difference. And then next one is CAR alone affects the percentage, right? So you can also just continue what we were saying, right? So compared to the control, always compared to the control, yeah? Okay, so CAR alone have maximum seed germination rate at around 95% looks like to me, right? Okay, whereas control max out around 80% germination, right? So again, I'm using this, yeah? Okay, so that would be the CAR, and I'm looking at TMB. So now it's just the same treatment. You look at the TMB. So this one, I want you to quote figure 1. So use the numbers from figure 1 because you're asked to support using data. So the data, you got to quote the data, right? So then now I'm doing TMB. TMB delays, okay, delays time, germination to around, so TMB alone, that'll be 21 hours, where control control germination germinates around, what do we say, 15 hours, right? So that would be the effect. It does affects that, right? Okay. And then the percentage as well. So TMB, okay, have a max, have maximum germination around 15 or 20, right? 15% compared to control 80%. So so that means lowers, TMB lowers percent of C germination. Yeah. All right, so something like this, okay? So TMB lowers the percent of C germination and have a maximum germination around 15% compared to that of the control at 80%. Okay, so just kind of polish your sentences. I didn't really polish all of them. So something like that would be fine. So that's part A. And if we go back to maybe the scoring guide, let's see what they say. Okay, and then we move on. So this should be 1 point for each bullet. Yeah, 1 point for each bullet. Yeah, so they didn't even quote the percentages, which is good. Yeah, I mean, so TMB, your germination starts sooner, higher percentage for TMB in the presence of only CAR. You should make reference to control. So I don't, but anyway, this is the bare minimum. Okay, so you should write a little bit more than just that. Okay. So 1 point each, please. So this 4 point total. So now the second part, let's see what they want for the second part, okay? Second part, let's take a look at this. This is the data. Okay, so the second part, let me copy the question so we can do it side by side again. So part B, we need to make a claim. Make a claim. So you have to write a claim. What is the claim, right? And it seems like here, let me move this a little bit. Oh, get rid of this. All right, there we go. Sorry. Make a claim. So here's my copy and paste stuff. All right, part B. Might as well do part C. So much time to do this on laptop. All right. So, and this. Here's my notes. Make a copy of this so I can do it side by side. So here it is. So what is the claim? If you start reading the question, it seems like yikes, this is not the right one. Here. So about rinsing. Now remember, rinsing is with water. You rinse with water. That's what they're saying. Yeah. So make a claim about rinsing. So if I remember earlier, the question says that both of these compounds are water soluble, so you can actually wash them away. That's what water soluble is. If they're insoluble in water like oil, you can't really wash it away. That's why you need soap, right? Your soap kind of help us dissolve the oil so that grease gets carried away. Nonpolar dissolves in nonpolar, right? So right now we're talking about that, okay? And then I need a graph not graph. I need the data. So here's the data. Okay, and let's look at what we are asked to do. So making a claim about the effect of rinsing of CAR to the receptor of the seed. T and D are chemical from the smoke goes on the to the seeds. So the idea is that if you wash water, maybe you can wash them off. So it's like removing the effect of CAR. What do? You wash it with water, right? And if you want to remove the effect of T and B, you wash away with water. Water allows you to wash them off. So you wanted the effect of rinsing with water on binding a CAR to the C receptor and the effect of rinsing on binding of T and B to the receptor. Identify the appropriate treatment groups and results from the table that, when compared with the control, provide support for each claim. So you have two claims. One is the rinsing on binding CAR. Why don't we focus on that? Identify the treatment group. So you see the treatment group here. I have 6 treatment groups on the left hand side. So I have treatment group 1, 2, 3, 4. So if I'm talking about CAR, okay, and rinsing, right? So rinsing on CAR. So I need to have CAR right here, number 2. I have CAR. There's no rinsing, no water. And then I have 5. I have CAR, right? And there's rinsing. So these 2 I have to mention, isn't it? And I also need to have a control for them. So the control is no CAR. Yeah. So okay. So let's see. Okay. So okay. So it seems like just from these 2, yeah, because the only difference is rinsing with water, right? Both data shows results different from control, right? You can see that. That means that whether you have water or you don't, whether you rinse or not, you get the same result that is different from the control, whatever that means, right? So it's not the same as the control. So that's the first thing that kind of popped to mind. So let's look at 1 and 2 are supposed to compare to each other. So you can see that 1 and 2 tells you that with CAR, you get different results from the control. And also 4 and 5, you can see from 4 and 5 tells you that the presence of CAR also shows you different result from control, right? So I don't know how many control I have to talk about because it says here appropriate treatment groups and the result from the table that when compared with the control. Okay, so maybe I don't have to talk about control specifically because they're saying compare with the control. I'm not sure. So let's just start writing them and see what we get. Yeah. So what would you guys write here? Because this is a little bit, isn't a little bit kind of annoying, right? Okay. Is this part? What is part B? Okay. So part B. Let's see what we can write here. Yeah. So part B. So it's a little bit small because we need the rooms here. Okay, part B. So what do you guys think? Here, rinsing. Yeah. I'm gonna go and say that rinsing has no effect or little effect because I'm looking at rinsing and to me they look the same. The results, right? Rinsing has no effect. So if you add water to that has no effect. Yeah, rinsing has no effect. Okay. Why does it keep doing this? Okay, I'm gonna get in the text. This is old versions. Okay, so maybe like this. Okay, there we go. Okay, so rinsing has no effect. On binding of CAR to receptor, right? Binding CAR to seed receptor. And I say no effect because I am looking at the data. Whether there's water or not, I get the same observation. Yeah. So again, I was looking at 2 and 5. Again, 2 and 5. The only difference is that they both have car, no TMB, and one without water, one with water. So I need to focus on 2 and 5. Okay. Okay. So no effect on 2 and 5. Okay. Both treatment group 2 and treatment group 5 produced results different from control. I think this would be enough. You don't have to write others, but so let's look at the scoring guide to see how are we going to get points for this. Okay, so I think that's enough. Otherwise, it become too wordy. I might be wrong. Let's see. Okay, so here's the scoring guide. Claim, yeah. So we did produce the claim right here. Rinsing does not affect CAR binding to the C receptor. That's the first thing I said, because our goal is to make a claim. You can also say that CAR remains bound after rinsing. Either one, because the effect is still there, right? So your support, okay. So it's only 1 point for the support. I'm trying to read here, right? 1 point for CAR. Okay, so yeah, we're looking at 2.5 on 1.4. Okay, so they're being pretty specific. I was looking at 2 and 5, right? Both produce result different from control. 2.5 on 1.4. I don't know if you have to be so wordy. I don't like that. So again, they're saying 2.5. So again, they're pointing out the one that we were pointing out, and then they try to mention both. But it's very wordy to do that. They're saying 2.5, 2 and 5 both produced. This is the control, right? And 4, yeah. So they produce different results from the control, yeah. So that's what they're saying. I mean, how do you put that into one sentence is quite difficult. I just said that. What did I say? I forgot. Okay, so let's come back to mine. Rinsing has no effect on binding. Both T2 and T5 produce enough. Okay, so from their controls, how about that? So if I put their controls, I think that would be good enough. Okay, and if you can say that T2 controls T1 and T5's controls T4 respectively. I think it's just too much. I think you can just stop at the control. Okay, yeah. Okay, so there's that. All right, let's look at the next one. Now we have to make claims about TMB and rinsing now. Okay, is there any questions? So if you have questions, just stop me anytime. I'm just blabbering. Yeah. Okay, so we're still doing the same. Now we have to look at effect of rinsing on the binding of TMB to the C receptor. So let's look at the results here. Okay, so I need to focus on TMB. So there's TMB. So again, I can cut off here, right? So again, that would be T3. TMB, no water, and 6. That's TMB with water, right? So do you see I get different result now? With water, there's no more effect. TMB has no more effect. It's the same as control. And then again, the control is T4. T4 is the control. So when you rinse seeds that initially were bound to TMB, seems like TMB gets washed off. So you get the same result as if no TMB, which is a control, right? Control has no TMB. Okay. But that's not the case when there's no rinsing. You can see that. So with the TMB, no rinse right here, you do get different result from the control. Control is 1, yeah? So I need to somehow put that together and make reference to these, yeah? So it's not a fun thing to do. We'll see. Let's see what we say. So let's continue. Rinsing negates the effect or renders TMB no effect. Rinsing removes the effect of TMB. removes the effect of TMB. Yeah. Okay, and then I need to make some reference. Yeah. Without rinsing or without water, TMB shows different results from control, and that will be T3 versus T1. T1 is a control, yeah. So let me just put that there, yeah. But when but after rinsing, T and B produce the same result. Okay, T and B produced the same results on germination. Yikes! As the control. So I'm looking at T6, which is with water, versus T4, which is your control rinse. Yeah. So they now have the same result. Again, that would be the focus right here. If I use water now, okay, water is used. I have the same result as the control, whether I have TMB or no TMB. So I'm using 6 and 4. Yeah. Okay. So I think that should be good. So let's look at the scoring guy. When they do this, yeah, similar to what we said, easier with the chart, TMB without rinse is different than control. That means, yeah, there's an effect, right? But TMB with rinse is the same as the control. That means there's no more effect if you rinse. Yeah, so that's basically what we said. So that means that rinsing gets rid of the TMB binding. So did we say that? I forgot. I think we did. So here, yeah, rinsing removes the effect of TMB binding. That's your claim. Yeah. Okay. So good. So this is 4 points: 2 for TMB and 2 for CAR. And make sure you make reference to this. This would be the hardest one because you always have to talk about control, guys. So always remember, given a control, make sure you use the control to justify your experimental group. That's what we are doing here. And it takes a little practice to get it right. Alrighty, that's that one. Let's one last one. There's an intense competition by plants to successfully colonize area that has been recently cleared by fire. So I guess the reason is why. So why do we have TMB and why do we have CAR, right? So we're going to describe one advantage of CAR regulation and also one advantage of TMB. How can TMB be an advantage if it inhibits and delays germination? We know that obviously the advantage for CAR is easier to talk about because you wanted greater germination sooner to utilize the newly disrupted area by fire, right? So CAR allows plant to germinate. And you want to take one step further. Yeah, that is the advantage. But so what does that do? So what is a big deal, right? To begin, for example, right? Colonize or utilize the newly opened habitat, right? Or opened area. Okay, this allows plant to be more competitive, right? Allow plants to have a competitive edge over plants that don't have CAR, don't respond to CAR, over plants that do not respond to CAR. Okay, so I'm trying to be a little bit more specific to describe the real advantage. Yeah, it is to germinate earlier. So what? So that we can utilize the resources sooner, so they're more competitive over plants that do not respond to CAR. Okay, so I feel that would be the most complete response. And then now we're going to advantage of TMB. So how can it be advantageous to a plant when TMB delays germination and has lower germination rate? When do you want to inhibit germination, right? So I'm thinking that what if the environment is not ready? What if it's still too hot? What if it's still too dry? What if it's not enough water? The fire start going on, raging, right? And if you germinate in that kind of condition, the chances of survival will be extremely low. So in other words, if you can promote germination, you also can delay germination. That's what TMB does, right? So TMB delays germination of seeds until conditions are favorable. And how does that work? Water is important. Remember, TMB effect is removed with water. So when there's water present, the negative effect of TMB is removed. Don't you need water to germinate? Yes. So when water is there, the TMB effect is negated, is nullified. So plant can germinate, right? So that makes total sense, right? So TMB delay germination of seeds until conditions are favorable when water is present, right? And TMB effect is nullified. Okay. So something like that. So let's see what the scoring guide would allow. Okay. So again, it should be 1 point each for those one. Oh, so many things in here. So let's see. What is the advantage of CAR? So germinate at times of increasing resource availability. Plants with CAR can outcompete other plants. Germination occurs when fewer competitors are present. Okay, that makes sense. And what's the advantage of TMB? Plants with TMB do not germinate and maintain seed dormancy until enough water is available. Great. Yeah, that's a good one. And plants with TMB regulation do not germinate in dry. So that would be a bad environment to germinate into. All right. What did I do? Okay. So that'll be it. Okay. Let's look at the next one. So we're done with the most difficult one. Then now you only have 4 points questions left. Yeah. So 4-point questions are much easier to do. So you should relax. You somehow got through the tough questions. Okay. And these ones are Gibber-Allen. Okay. So again, you can see the first 2 questions. There's no background information that you can prepare yourself for. How do you prepare yourself for a question like CAR and TMB? You cannot. There is no such experiment in AP Bio textbook. So you're not trying to memorize the entire plant chapter. Your teacher at school doesn't even go over plants. But this is about survival, adaptation, right? This is about gene expression, about signal transduction. Something from smoke lands on the soil, and that affects how plant germinates, when it germinates, and also what % of the seed germinate to ensure survival. So, and if you look at question number 1, too, who would have known caffeine can improve memories of bees? And you got to design an experiment. There's no place that you can learn in AP Bio book that specifically addresses caffeine on bees. So you can't prepare 1 and 2 traditional ways. Memorizing a whole bunch of vocab is not going to help you. So make sure you understand that is the way AP Bio is tested. You have to do it on the spot, impromptu, right? Like those comedians who do impromptu. Yeah, you just have to have a bunch of stuff ready and ready for whatever. All right, so let's take a look at this one. Now, this is about Gibber-Allen, so that is a plant hormone. Okay, so what does it do? It tells you what it does, so you don't have to memorize it. There's no point of memorizing that. Gibber-Allen promotes stem elongation. Yeah. So, for example, if you're a cabbage is actually stem. I don't know if you never noticed. Okay, you're eating the leaves. It's a very short, stubby stem. If you give cabbage a bunch of Gibber-Allen, you're gonna get a big, long cabbage. It's very weird looking. Yeah. So that's what stem elongation is. And then why is it important for plants to elongate the stem so that they can be tall? The taller they are, the more sunlight they will have, the more competitiveness for sunlight means that the more growth they can support themselves with. So it is important for plants to be tall, right? Unless you're shady plants, right? So that's what this hormone does. Okay, what about it? So Gibber-Allen is the hormone. I'll say GA3H, so don't try to pronounce the word. So GA3H is the enzyme that catalyzes the reaction that converts a precursor of gibberellin into the active gibberellin. So the way this interpret to me is that yes, GAP, yes, active gibberellin. No GA3H, no gibberellin, right? You need to have GA3H in order to get active gibberellin. Otherwise, gibberellin stays at an inactive precursor form, which is useless. Okay, so you need an enzyme for gibberellin. All right, a mutation in the gene for this enzyme, so DNA to RNA to protein, since that's what they're going after. So these mutations can result in short. Of course, if you are mutated without this enzyme, without active gibberellin, you don't have stem elongation, right? So you are short. So if you're mutant, you're short. While pure breeding tall is crossed with pure breeding short, all F1 are tall. So what are they telling you? Tall is dominant and short is recessive. That's what they're trying to tell us. Okay, so right away, they're trying to connect this to genetics. And tall will be no mutation. That's why they call them wild type. Wild type is the most common. So wild type, okay? The most common phenotype is tall, there's no mutation. So the short one must be mutant, yeah? So if you have a mutant allele, like that, okay? So this would be the parent generation, tall, crossed with short, and the F1 is all T, little t, all tall. And then it says, when F1 are crossed with each other, so they're not doing a test cross, they're doing F1 cross with each other. That's F2. So when F1 crosses with F1, two heterozygous tall cross with each other, you get 3-to-1 ratio. That's Punnett square, basic Punnett square, right? So 70% are tall, that's 3-to-1, 25% are short. So they tell you these information. Okay, so what about it, right? And they give you a codon table. Remember, codon table is translation of codon into amino acid. This is the job done by ribosome. And there's always the start codon, which is methionine. You can see that on the bottom. This AUG is always the first amino acid whenever proteins are made. They will be cut out later on by rough ER as they modify through the pathway. And there's 3 stop codon, UAA, UAG, UGA. So out of 64 codons, 3 are stop codon and 1 is a start codon. And genetic codes are kind of redundant. You see that all these codes for alanine, you see that? So this means that you can change the last nucleotide. The last nucleotide can be UCAG, as long as the first one is GC, then you have alanine. So genetic code is redundant, yeah? Not repetitive, it's called redundancy. Okay, part A. So again, these are 4 points all in all. So they're pretty short questions to answer. So let me just take a picture of this so we can start answering these, yeah? Okay, so again, you need gibberellin to become tall, to elongate the stem. And the enzyme GA3H is responsible for getting you active gibberellin. If they're mutated, no gibberellin, right? So a mutant cannot produce gibberellin, so the phenotype is going to be short. And with the active enzyme, then you're tall, okay? So this is question number 3. Let's say, how do we answer this question? Yeah, let's take a look at this. So question number 3. All right, the wild type allele codes for enzyme. Okay, so the wild type is the dominant T, codes for enzyme with alanine, which is a nonpolar amino acid. So we're told that one of them, alanine, is nonpolar. Wild type is nonpolar. Okay, and then at position 229, so the big long protein, the 229 amino acid codes for, it's actually an alanine. The mutant allele, so the mutant one is little t. Okay, the mutant one is little t, right? So they're mutated. So they're short. And they code an enzyme with threonine, yeah? So if you have threonine, the enzyme doesn't work. You cannot get active gibberellin. Okay, so that's what that is. And alanine, you can't, right? So, and this is a polar, so that's the difference. So when the non-polar one became a polar amino acid, it renders the enzyme non-functional. Describe the effect of the mutation. This is called the substitution, right? So remember, there's a point mutation. When we talk about the point mutation, that means that one single nucleotide. And you can have substitution, which is this one here. Okay. So in this case, alanine becomes threonine. And when you are threonine, the enzyme is not active, and you don't get active gibberellin. You are short. So that's substitution. You can also have deletion. You can also have insertion, and deletion, insertion can cause very bad, what is that called? Frame shift, right? Frame shift mutation get a whole bunch of errors, right, from the point of insertion or deletion. All right, so describe the effect of the mutation, provide reasoning to show support how this mutation result in short phenotype in homozygous recessive little t. So that's not too bad, right? We already kind of discussed all the implications of not having gibberellin. So, VA, what do you guys think? Okay, was important to talk about this. So when I do this question, I have to read the question several times because sometimes there's so much stuff going on. I don't remember what they want. Do they want to describe? Yeah, they want to describe what, right? They want to describe the effect of the mutation. So the effect of the mutation is to render the enzyme non-functional and provide reasoning. So reasoning. What is the reasoning that this mutation give you short plant? Yeah. So come back to this again. So the mutation renders the enzyme. I'm just going to say GA3H enzyme non-functional. Now, functional, the function of enzyme depends on the three-dimensional structure. So do I need to talk about that, right? If you remember, I would, because you can argue that's part of the description, right? So how does it become non-functional? Because that polar amino acid, right, disrupt the 3D structure. So no 3D structure, no enzyme function, right? So I want to add that. So the mutation renders the G3 enzyme non-functional, okay? Because the tertiary, right, tertiary 3D, tertiary structure or 3D structure of the enzyme is changed as a result of the substitution. Yeah. So again, that's more detail than we need, I believe. Yeah. And now that is the description part, right? The effect of mutation on the enzyme. Did I describe that? That the effect of mutation render it non-functional because it changes the tertiary structure. Yeah, I did that. Now provide a reasoning to support. What are we supporting? How the mutation results in a short. So without the active, well, without the functional enzyme, functional GA3H, no active gibberellin. Gibberellin is formed. No stem elongation, short phenotype. No stem elongation would result and results in short plant. Yeah. So the connection is to gibberellin, and we knew this because in the reading we're told that was the case. Yeah. And if you look at the reading one more time, we don't know this unless we remember what we read, right? So it says one more time here that gibberellin, that's how you spell it, 2B, promotes stem elongation. And this enzyme that catalyzes the conversion of a precursor to the active gibberellin. So you need the enzyme to do it. So if the enzyme is mutated, then you do not have gibberellin in the active form, then you don't have stem elongation, therefore you get a short one. And that's what we are told. So they just want you to connect them together. All the informations are here. So feel free always to come back and read those detailed questions when all the information that you need to answer the questions are already in there. Yeah. So try to fish out in a logical way. All right. And in part B, use the codon chart. Predict the change in the codon sequence that results in the substitution of alanine or threonine. Yeah. So alanine to threonine. So let me get rid of this. So part B, we have alanine becomes threonine at the amino acid position 229. So amino acid 1 is 2, then 3, then 4. This is a big long protein. Okay, gibberellin is a protein. Yeah. So at this position, so you look at the codon table, and that's your answer. So I'm looking for alanine to threonine. So right here. So alanine here, threonine is right above it. So what's the difference between alanine and threonine is the first set of nucleotides. If you are A, then you're threonine. If you are G, then you're alanine. So this is a substitution of G become changed to A. So you'll change alanine to threonine. G to A in the first codon position. This is not the third. The most left one is the first codon position. Okay, so G to A substitution in the first base of the codon. So let me go back and write that. So this would be a G to A base substitution in, and you have to say which position, yeah? Do I have to do a generalized or no? Okay, so it says using the codon chart, describe the change in the codon sequence results in, yeah, G to A base substitution in the first nucleotide position of alanine. Okay, so in the first nucleotide position of alanine, G is changed to A, and that's what this is, right? So make sure we answer the question. Did we answer the question? Okay. So give me a sec. Okay, yep. We did answer the question, right? Predict the change in the codon sequence that results in the substitution of alanine to threonine. Yep, we did. Okay, good. So we answered that. And last one. Okay. The question is, let me get rid of this thing here. Clear? Okay. So we're trying to answer the question how individuals with 1 heterozygous, right, big T, little t, or 2 homozygous copy of the wild type. So again, they're saying that we're talking about big T, which is that one, homozygous wild type, and big T, little t. Yeah. So can have the same phenotype. Well, they can have the same phenotype, right? Big T, and big T, little t can have the same phenotype, which means they're tall. Because in heterozygotes, right? In heterozygotes, the wild type allele, the normal allele, big T, the normal big T allele produces enough enzyme. Even though little t cannot, right? Produces enough GA3H enzyme to produce enough enzyme to have enough gibberellin to be tall. Kind of wordy. Let's see. So the key is that how come heterozygous basically can also be tall, right? So because they produce enough enzyme to have gibberellin to be tall. A little bit wordy. I don't like this, but okay, something like that will be fine. Okay, so it doesn't have to be perfect, but as long as the grader knows that you're not ESL, so try to do try to write it simple sentences. You can use several sentences. I would polish this a little bit. Yeah. Okay. So I would say that the dominant allele compensates for the recessive allele. That's another way to say it. So that the plants have enough active gibberellin to be tall. That's what I'm trying to say. Yeah. Okay. So it's hard to write that out. Okay. So let me just write it again. So if I were to correct myself, part C. Okay. I will be saying something a little bit different. And then we're done with this question. Yeah. Oh, my writing is so big. Such a baby, little tiny C here. Okay, so here, let's say that the T allele in the heterozygotes allows the production of enough active gibberellin to be okay. So something like that. Okay. So it's a little bit different from what I said earlier. Also. All right. That's that. So these points are easy to do. See, every bold letter is 1 point. So please give yourself 2 points for A, 1 point for B, and the maximum 1 point for C. Okay, let's see what the scoring guy said. Mine as well. Okay, so this one, so part C, they say enough active enzyme is produced from one dominant allele. Okay, that's great. That's much more concise. Enough gibberellin is produced in the presence of just one dominant allele in the heterozygotes, right? Okay, so that'll be that. Let's look at the next one, please. So number 4, again, those are 4 points question. So dietary composition of organism in an aquatic ecosystem. Yeah. So we have algae, different organisms. So how come algae has no diet? You should know that algae are photosynthetic. They don't eat other things. So that is the base of the food chain. So everybody will be feeding on algae. But algae does not have a food diet. It does not eat any of them, right? So it's photosynthetic. It's producer, autotroph. We have stoneflies, midges, hellgrammites, and caterfish. Now, if you've never seen hellgrammites before, they're pretty disgusting, yeah? They're big. If you see them, if you're a bug, hellgrammite, how do you spell hellgrammite? If you're a bug, you want to stay away from hellgrammite. They look nasty, yeah. The bug right here, insect. Okay. Yeah, they like hellgrammite. This is a kind of scary looking thing. Yeah, you want to stay away from this. Yeah. Okay. So that's probably the top of the food chain. Yeah. Okay. So anyways, if you look at this one, okay. So how do you read this? Well, you got to figure out the orders in the trophic level. Or this is could be a food chain or food web. We don't know. But you can see that midges, the food is 100% algae. So they eat algae. So this must be the primary producer. Right? See that? That's important. And stonefly eat midges and also caddisflies. And caddisfly eats algae and midges. And hellgrammites eat everybody. It doesn't eat algae. So it's a carnivore. Right? It eats stoneflies, midges, and caddisfish, right? Like that. Okay. So anyway, so there's a lot of relation there. So the question is that the table above shows how much each organism in an aquatic ecosystem rely on various food. The row represents organism in the ecosystem, the column is the food source, the percentage, blah, blah, blah. So we already read that. Okay, so based on the food source indicated, construct food web template below, writing the name of the organisms and draw the arrows. You draw to the guy that eats you, right? Okay. So there's that. Make a template and then use the table. We're able to figure out this. This looks pretty easy to do. You just have to double check And if you're not sure what to put in there, just put something there and then go with it. And then if you find any conflict information from the data, then you just readjust. So don't be afraid to do that. Yeah. The key is that once you start putting stuff down, things become more clearly. And then you can see if your proposed answer conflicts any of the data. So don't be afraid to do that. The worst thing is just to stare at it for like 2 minutes and then start thinking about Disney movies. Yeah. Okay, that's the worst thing. I heard one of the way to jeopardize your classmates, whoever taking the test with you, is to sing a Disney song in the beginning of the class. And then that will resonate in their mind for like the rest of the test. Yeah. And that was a horrible way to do to your friend. Yeah. So even if that happened to yourself, you self-jeopardize yourself, you still have to overcome that. Yeah. Start singing Disney songs. They brainwash us so that we think Disney is the happiest land on earth. So anyway, that's a brainwashing right there. Okay. All right. So let's look at this one. So again, bottom of the food chain must be the algae because they're not eating anyone. They're the producer. So they eat by midges. So we take care of that already. So that's not too bad. Okay. They're eaten by midges, right? Okay. And then want to look at midges. So we know the midges right here, they're 100% algae. So we already took care of that. So midges are eaten by everybody. So let's see. So how do we decide? I'm looking at a stonefly. 90% is midge and caddisfly. And also I'm looking at caddisflies eat algae and midges. So this must be caddisfly then. Caddisfly, because caddisfly eats midges, which we have, and 70% from algae. So there, that's 70%. So this is like 30%. This is like 70%, right? If you want, you don't write in the numbers. I'm just writing it for analysis. And then I have stonefly then, right? So this must be stonefly. Stoneflies eat midges, so 90% of the food come from midges, and almost 100% midge, right? And then caddisfly, so I got to draw an arrow right there. So that's like 10%, right? And the last one, the top of the food chain is the ugly thing, the healthy, yeah. So then it's stonefly, so stonefly have to go to it. That's 20%, and midges is 10, and finally 70% is caddisfly. So I need a arrow from caddisfly. That's 70%, right? All right. Again, don't put the numbers in. Okay, don't put the numbers in. There's no point of putting the numbers in. I'm just putting it there. Yeah. All right. So that's it. Construct a food web in the template. We did. Draw arrows. We did. And then you're done. So that's like at least 2 points because you have 2 points left. Okay. Copy. In effort to control the number of midges, an area. So you want to control midges. Why do you want to control midges, guys? Can you give me a reason why midges population must be controlled? If it's not controlled, if the midges are overpopulating the ecosystem, it's going to destroy the algae. That's the problem. Okay, because 100% feeding on algae. So that's why you got to control the population. Yeah. Okay. And which one would be a keystone predator the best, guys? If you want to control midges, who is the best keystone predator? That means the predator that feeds on midges and keep it under control. Probably, if you guess, it's probably the stonefly. That would be keystone. It's most responsible for keeping midges under control. If you had to guess, right? I'm just trying to come up with some additional questions ecology can ask you, right? Remember, the keystone predator is the one that maintains biodiversity. Without the keystone predator, like stonefly, midges will go out of control and destroy the bottom of the food chain for everybody, which is not good. So keystone predator is what that means. So if I had to guess, the keystone predator would be stonefly, since 90% of its food source is the midges, yeah? Okay, so the question here, we are spraying with fungi to keep midges under control. Now this will decrease the midge population. Based on the data, predict whether spraying the fungi have the greatest short-term impact on the population of stonefly, caddisfish, and helmet rat. So what would this affect the most, right? So if you spare the fungi and kill the midges, then stonefly will be mostly affected, right? Okay, isn't it, right? Because stonefly depends most heavily on midges, as 90% of the food source are midges, right? And that's your justification. And that's it. Yeah. So let's try to put that in words ourselves. Again, practice it in your head, practice writing it down. If you're not sure what to write down something so you have something to look at and build upon. B. So predict your prediction, right? So now I guess it's a good idea to write out the numbers because it allows you to see the answer more clearly. Yeah. So let's see. So this one I'm gonna put. What am I saying here? So what did I say? Stonefly. Okay. So predict, right? So I don't have to say anything. Justify. Okay. 90% of stoneflies nutrition midges. It has the greatest dependence on midges. Midges? I mean, midges. How do you spell midges? Midges, yeah. And any other, well, I could say what they are. Let me just say, then doesn't take too much time. Then caddy flies or and helmgramite. Okay, like that. All right. So justification and your answer, easy, right? So that's easy ecology. You don't have to know so much about ecology to answer that question. Let's look at the next one, please. Next one is this one here. So this is also 4 points. The old AP has 8 questions. The new one only has 6 questions. So let's look at this chart. We have 3 graphs, and the x-axis is time. As time progresses, and we're looking at the concentration of oxygen, cyanobacteria. Well, first of all, cyanobacteria is photosynthetic. Okay, you should know that, which is why there's oxygen, right? And decomposers are like bacteria and fungi. Those are the 2 major decomposers. If you had to squeeze another kingdom, bacteria, fungi, and also protists, right, are good decomposers, but definitely bacteria and fungi. And decomposers, they are not photosynthetic, so they will use the oxygen by cell respiration. And that's what the question is saying. Okay, so don't overanalyze the graph right now. You have different time. So you have this freshwater photosynthetic cyanobacterium. So again, don't try to pronounce that. I don't even read it. So you have photosynthetic cyanobacteria. That's the cyanobacteria right here. Yeah. Okay. When temperature increases, nutrients are readily available in the pond. All right. So high temperature, lots of nutrient, right? So they're light temperature, right? This thing here will undergo rapid cell division and forms large visible algal bloom. Now, algal bloom is not a good thing. So when the environment or lake or rivers have algal bloom, it will exhaust all the oxygen in the lake. And that is not good for everybody else dependent on the oxygen. And the reason is the decomposers, yeah? So when you have an algal bloom, when there's too much fertilizer in the nutrient, for example, too much nitrate and phosphate from the runoff, from the neighboring fertilized farmland, you're gonna get algal bloom because these cyanobacteria grow so quickly, they're gonna cover up the surface and they're gonna die, and then the decomposers is gonna consume all the oxygen. Look at oxygen, how fast it drops. So when the decomposers start to rise, when the decomposers population, you can see that from the chart here, when the decomposer population start to rise, the amount of oxygen dropped precipitously. And when there's no oxygen in the water, then larger consumers like fish, other fish, and more fish, they start to die, right? So that destroys the entire ecosystem with algal bloom. Algal bloom is not a good thing, yeah? Okay, And the question is, identify the metabolic pathway and organisms that is primarily responsible for changing oxygen between 1 and 2 and 2, 3 and 4. Now, do I need to know algal bloom, whether it's bad or not? Maybe not, because to answer this question, between 1 and 2, I just focus on oxygen level. Oxygen level is increasing between 1 and 2. And then only metabolic pathway and the organism, it has to be photosynthesis and cyanobacteria. That's it. So you don't need to know algal bloom. You need to know photosynthesis by cyanobacteria between 1 and 2 is responsible for releasing oxygen. That's it. That's all you need to know. And then between 3 and 4, oxygen level drops. And not because cyanobacteria, because they're dead, right? So it must be decomposers. And what process use oxygen? Aerobic respiration, right? Cell respiration, aerobic respiration. So that's like no-brainer here. Easy points. So 4 points here, 1 for photosynthesis, 1 for cyanobacteria, 1 for decomposers, and 1 for aerobic respiration. Write down 4 words. You have to justify? No, it says identify. So don't waste time on this, but identify. So write down the process and move on. So we would save a bunch of minutes on this. Okay, so 4 words, 4 points. All right, let's look at the next one. Okay, identify, I love that the best because there's no answer. Or calculate, identify, state. No justification required. All right, let's look at the next one. Next one, we don't know what's going on here, but it describes that process for us. So let me take a picture of this, or maybe I can write on this. This is called a comment assay. Yeah, assay is some kind of test. Okay, comment assay. What is it for? No, I'm actually going to. Okay, let's just read it here for now. So the comment assay is a technique used to determine the amount of double-strand breaks in DNA. So it determines DNA damage. Now, what is double-stranded break? Remember the DNA is double-stranded, double helix. So we're not talking about the breaking hydrogen bond. I think we're talking about the breakage between nucleotide that run 5 to 3, 3 to 5, anti-parallel fashion. Yeah. So when the double-stranded DNA break, we're not talking about separating TAGC base pairing, not the hydrogen bond, but it's covalent bond. So that would be a DNA damage. Because if you separate the bases, they can come back again. They can anneal back again. That's called re-annealing. And then there's no DNA damage. But if you break the strand, the sugar phosphate backbone is broken, then you got a real DNA damage. And those DNA damage should be destroyed. Those cells with DNA damage should not continue and do cell cycle. Yeah. All right. So talk about this one. So what does it do? Comment assay. Assay is to detect DNA damage. Okay. The nucleus of a cell is placed. So you put a nucleus here. Right here. You put a nucleus there on a microscope slide coated with agarose, like a jello stuff. And electric current is applied to the gel that causes the DNA to move. Now, electrophoresis, we talked about this term before. It is to separate the DNA based on charge and size. So can you tell me here, would you agree I have to put positive terminal and negative terminal in this fashion for the DNA to migrate to the right? Because DNA is negatively charged because of the phosphate. DNA is sugar, phosphate, nitrogenous base. Phosphates are very negatively charged. So this is a separation by size and charge. Smaller fragments will move faster. I guess that's the tail one, right? Wouldn't that mean that the fragmented one are a little smaller than the double helix, which are larger? So smaller fragments move further down than larger fragments. So separation by size of the fragment and separation by charge. So you have to put positive terminal on the right side for it to move towards the right side. If you revert the terminals, you're not going to see separation. Gel electrophoresis, phoresis means to separate by size and charge. And DNA is scanned. So if you don't scan the DNA, you're not going to see anything. DNA does not have color, it's transparent. So you got to scan it. When viewed using a microscope, undamaged DNA appears as round, which is the head. the fragment of the damaged DNA is smaller, so they will appear as a tail. It looks like a comet, right? If you're lucky enough to see a comet, it's awesome. Yeah. Okay. I saw it when I was 7 years old. What is that? Halley comet. That will come another 70 some years. So I have to wait until if I'm 80 something to see it again. So, but that was a comet. Yeah. So, and you'll remember. So that's that one. And the length of the tail is the amount of damage. So that's also important information because that surprised us, right? The length of the tail, amount of damage. That makes sense. The more damages, the more fragments, the longer the tail, right? Again, the tails are smaller fragment. That's why it moves faster towards the positive terminal in the first place. So hopefully this common essay is something that makes sense, yeah, using gel electrophoresis. The damaged DNA are smaller fragment that will move down farther, faster. Okay, then the undamaged one. To explain the movement of DNA fragment in the common essay, identify one property of DNA and provide reasoning to support how the property contributes to the movement of common essay. Yeah, so we talked about two properties, size and charge, right? So size and charge explain the fragment of the DNA. Provide reasoning, okay, to support how the property contribute to movement. So, so this is number 5. Okay, size and charge. Okay, so you can talk about either one. Don't talk about both. They're just one property. So it depends which one you like. Okay, so why don't we talk about the charge first? Yeah. So DNA molecules are negatively charged. So that's one property. You can put a period there to answer that question. Yeah. DNA molecule negatively charged. Period. So that's the identification. We have identified that property. And now you got to explain reasoning, right? Support how the property contributes to movement during this. Yeah. So this positive terminal pulls DNA, okay, and through the agarose gel, resulting in DNA movement. Yeah. So if you talk about the size, DNA damaged and undamaged DNA have different sizes. So the size, yeah, they have different sizes. Okay. I thought I typed it out. They have different. Okay, different sizes. Okay, so the damaged DNA, which is the tail, right? Smaller in size and moves faster to the gel. So smaller fragment move faster. So either one, don't put both, but whichever you want. Yeah. Okay, so that's that one, and that's actually part A. They go all part A. Yeah. So this is OR, not part B. So next one is part B. What about part B? Yeah. Part B says in a different experiment, cell treated with a chemical mutagen that cause only nucleotide substitution. Now remember this one in the first sentence, how this comet assay works is that you have to have a double-stranded breaks. If you have a nucleotide substitution, your double-stranded DNAs are intact. There's no fragments. So predict the likely result. There will be no tail. That's it. Yeah. So no tail. So let's look at the scoring guide and see how much they'll give you points for. So again, this is just 4 points. Okay. So let's move on. No tail. No tail. I don't know where I am. Okay, so this one right here. So let's see what they give you. DNA have negative charge, DNA have different sizes, right? That's it. And then, so no tail, or tail will be shorter. So either one, that's your prediction. You can say head only, or you can say there's no tail, or you can say the tail will be shorter. I don't think we should say shorter, just say no tail. So they'll take any answer, basically. Yeah. All right. So 1 point for the prediction. So this question is question number 6. Okay, this is only 3 points on the AP test, yeah, old AP. So the new AP does not have any more 3-point questions. So 6, 7, 8 are 3 points each. So question, yeah, 3 points. 7, okay, we're almost there. Okay, number 7. Where's number 7? Right here. Okay, so this one does not have diagram. Which is not going to be there anymore. Okay, you're always going to have diagrams and graphs. That's my guess. Yeah, but this is the first year. All right. So many species of bacteria grow in mouth of animal for sure. That's why you have to brush your teeth. Actually, brushing teeth is not so good. You should floss. That's the best. Brushing teeth doesn't work very well. Floss and rinse your mouth, then brush your teeth. All right. So anyway, so you have biofilm, which is a teeth plaque. Yeah, that's when you go do a teeth wash, the doctor will use a pick to break apart all the plaque, which is actually a film of bacteria around your teeth. Yeah. So, and within that is the enamel. The surface of the tooth is enamel. If they puncture the enamel, they will go into the dentine, where your nerves are, where nutrients are. That's the goal. And they will make a hole in the dentine. That's why it's called a cavity. Tooth cavity is basically a hole through the enamel, and they live in your dentine. Okay, so that's what that is. Yeah. Okay, so outer layer contain high level of oxygen, makes sense. It's exposed, and the closest to the tooth has low level of oxygen. And the bacteria can use the oxygen. Some of them are anaerobic, some of them are aerobic, I guess. Yeah. So they tell you what enamel is, which can be dissolved in acidic condition. When enamels break, the bacteria can extract nutrients and tooth and causing cavity. Certain type of bacteria, so you have this one here, mutans, so S mutant, right, live in the innermost anaerobic, so they don't use oxygen, and associated with cavity, so they trigger cavity. Now you also have the sanguineous that compete with mutant, right, the competition here. They're not eating each other. You're told that this relationship is competition. But they're not able to thrive in acidic environment. So when the mouth is very acidic, you get cavity. Mutant flourishes. And okay, that's what they're saying. All right. So identify the biochemical pathway mutant uses for metabolizing sugar. Now, if these guys live in anaerobic innermost layer, then it has to be anaerobic respiration. Right? Anaerobic respiration or fermentation. There's 2 types, alcohol fermentation or lactic acid fermentation. So this would be lactic acid because of the low pH. That is the description, right? So when sugar is metabolized by lactic acid fermentation, lactic acid causes the pH to drop. And that kills the other guy, which has double benefits, right? All right? So that's the answer. And normal tooth brushing effectively remove much of the plaque from the flat surface, but cannot reach the bottom. Cannot reach the surface between teeth. That's why you floss. Tooth brushing is not effective. Many commercialized toothpaste contain alkaline component. Well, do you see that alkaline component? Well, is better for the other guy, which raises the pH. Predict how the population of S mutant and S sanguineous are likely to change when these toothpastes are used. So that means when the pH is higher, sanguineous population will go up, and mutant population will go down, right? That's basically it. Okay. Again, your clue is that mutants are unable to survive. Who are unable to survive? Sanguineous. Sanguineous are unable to survive in acidic. So, and they will outcompete this as they are surviving better in the alkaline solution. Yeah. So is that okay? So that's this one. Okay. We probably don't have to write this down. This is pretty self-explanatory. But let's just look at what the answer key says. Okay, so the answer key would, why? I thought I deleted it. Okay, let me see. Clear again. All right, so let's look at the answer key. Okay, so identify the biochemical pathway, so anaerobic respiration or fermentation. And then you should talk about lactic acid fermentation, yeah? You can even, for people who put glycolysis would also work, as long as you said that pyruvic acid is also acidic. So you don't have to go through fermentation. Pyruvic acid is a little bit acidic, that's fine too. So even that, okay? So lactic acid, all right. And next one, normal brushing, remove plaque, right? So if you use the toothpaste, what's gonna happen? Mutant will decrease and squeakiness will increases. So that's your prediction. Do you have to justify it? No, it just says prediction, yeah? But the reason is this alkaline condition makes the squeakiness more competitive as they're able to flourish now. Okay, before in acidic medium, they actually die, right? All right, let's look at the last one. Okay, last one, number 8. Okay, and number 8 right here. So let's talk about estrogen. Estrogen is female hormone. So this one I want to review quickly. You have 2 types of hormones in your body. One is made from lipid, and the other one is made from proteins. Lipid hormones and protein hormones, they both travel in blood to reach their final destination, yeah? However, the mechanism, how the 2 hormones work, very different, okay? Your protein hormones, for example, they do not enter the cell like lipid hormones, steroid hormones, okay? Because protein hormones are too big and polar. So they use signal transduction. So I need you to remember that, yeah? Okay, let me just type out something quick here. Okay, so this is the protein versus hormones versus lipid. Lipid hormone means steroid hormone, yeah? All the hormones in our body are protein, except the sex hormones, which is estrogen, testosterone, progesterone. So their mechanism are different, okay? Although both are released into the blood, protein hormones do not enter the cell, lipid hormone do. Protein hormone bind to membrane receptor because they don't enter. Steroid hormone bind to a receptor within the cytoplasm. So do not enter cell, okay? And the lipid hormone, they do, yeah? So let me stretch it out. Each one, lipid hormone, yikes. Lipid hormone do enter. And also, protein hormone use signal transduction. Okay. This one binds to membrane receptor. And these ones are intra or cytoplasmic receptor. How about that? Cytoplasmic receptor. It's a big difference, yeah? Okay, so their receptor is different. Protein hormone uses signal transduction. Hormone controls gene expression. They don't use signal transduction, yeah? Okay, so that's another big difference between them. So let's go to protein hormone. And remember, signal transduction has 3 stages. They use signal transduction, receptor binding, and secondary messenger, cascade of reactions, right? And cell response, yeah? And these ones, they bind to DNA. Once they enter and then bound to the cytoplasmic receptor, they bind to DNA. And increase gene expression. Okay, so they bind to DNA and increases gene expression. So don't forget the signal transduction has different stages, yeah? Okay, receptor binding is first one, okay? So the question is this, okay? One characteristic of plasma membrane that allows estrogen, nonpolar. Plasma membrane is a nonpolar barrier. Estrogen is nonpolar barrier. They can pass through the membrane because of that. Second, in a lab, researchers generate antibodies that bind to purified estrogen receptor extracted from the cell. So the idea is that they want to use the antibody to treat estrogen-dependent cancers. So they want to use antibody to bind to the, so what these antibodies can bind to estrogen receptor, and that will reduce cancer, yeah? Reduce cancer. Why? Because the actual estrogen are no longer allowed to bind if the receptors are all covered with antibodies, right? So that means you reduce cancer for that reason. Now, why is this ineffective? This is ineffective because the antibodies will not be able to access. Antibodies are also proteins. They are unable to enter the cell. The proteins, the big proteins, they can't enter the cell and reach the cytoplasmic estrogen receptors. So you can inject a bunch of antibody into the bloodstream, they will have no access to the cytoplasmic estrogen receptors in the first place. So it's not going to work, okay? So that's not going to work, but that's how antibody travels. Antibody also travels in your blood. So let's just look at the answer key and finish this one. Okay, so this one again, antibody is unable to enter the cell. They will not be able to bind to the intracellular or cytoplasmic estrogen receptors. Okay, all right, so that will be it. So again, guys, next week, I think test 5 may be multiple choice. I'm not sure. Yeah. So maybe the first time we do multiple choice. No, another free response. Yeah, we're almost done with some free response. All righty, guys, we'll stop here. I'll see you guys next week. Thanks, guys. Take care.